So, welcome everyone and good evening. Welcome to our interdisciplinary online seminar series, the second session with the topic of biomaterials. At this juncture, we are facing very complex various challenges in the world. So the world requires more effective and innovative solutions. As complex problems are often not suitable for single discipline research anymore, and also the boundaries of multiple scientific fields have been merged, the researchers need to broaden their horizon by investigating the various realms of expertise. I excuse in the middle because I have to admit the newcomers. Sorry. And therefore, in today's economic and social condition, the possession of a broad set of skills with high levels of adaptability seems to be an imperative for university graduate students in all different fields. Therefore, this seminar series with various topics have been organized by architecture faculty and the collaboration of Institute of Graduate Studies to familiarize students, postgraduate students, with the nature of interdisciplinary research and university research activities in various faculties. Our plan for this session, which is the second session of our seminar series, is we are very honored to be with Professor Dr. Terin Adalı and Dr. Marziye Geranpaych on today, the 12th of April 2021, the second session starting with the main topic of biomaterial. Dr. Terin will present a presentation with the title of Natural Biomaterials and Biomedical Applications. And Dr. Marziye will present a topic called The Future Belongs to Biomaterials. So before I give the room to our dear keynote speakers, I would like to introduce Professor Dr. Terin a little bit. Of course, our dear colleagues and faculty members know Dr. Terin very well. It's just a brief introduction. Dr. Terin is a lecturer at the Department of Biomedical Engineering at the nearest university, and she's currently the director of the Research Center of Biomaterials and Tissue Engineering Research Center at the nearest university, and also associated researcher in Sabancı University at Nanotechnology Research Center. She has supervised various and co-supervised several PhD and master theses and published articles in the international acknowledged scientific journals. Her current research areas include synthesis, characterization, and biocompatibility, studies of biomaterials from natural resources. And she is currently leading and contributing to too many research projects. And also I would like to introduce our second keynote speaker, Dr. Marziye Geranpaych from the Department of Nanobiotechnology at Terbiyat Modarres University, Tehran, Iran. Dr. Geranpaych has held a PhD from Terbiyat Modarres University, and she has spent her sabbatical leave in National Center for Nanoscience and Technology in China in 2018, where she's published many papers on cancer and metastasis. She's now a postdoctorate researcher in TMU University, working on increasing the stability of some enzymes and their delivery to different tissues. So I would like to ask Professor Dr. Terin to please present your topic. So the room is yours, Doctor. Thank you.
Yes, thank you so much. First of all, I would like to thank this kind invitation to present our works in Tissue Engineering and Research Center with my team. And it's an honor for me to present this presentation for the audience. And let me start. Yes, natural biomaterials. And of course, modification, processing of these natural biomaterials and the applications on the biomedical field. I want to start with the latest definition of the biomaterials. In 2018, European Biomaterials Society redefined biomaterials as materials that can be implanted within or used in conjunction with the body. So contacting of the biomaterials with the living tissues are very important. So they have to design to have properties or biomimic the properties that closely matching that of the biological system. This is very important and the newest definition for the biomaterials. Of course, they have to be stable depending on the application. As we go further, we will discuss what type of applications we are talking about in the biomedical field, of course. Bioactivity is also very important. They might be, depending on the application, bioabsorbable or biodegradable. So these properties are also very important to be a biomaterial. And of course, designing to partially and completely fulfill the functions of the disease, damage, or malfunctioning tissues and organs is very, very important. While we're trying to biomimicking any tissue or any organ, we have to take care of all these important properties of the biomaterial during the design. Of course, biological molecules that are naturally origin decellularize tissues. And this is very important. They are received, the resources of them might be plant, animal, bombox mori, insects, microorganisms, crustacean, and algae, and also there are other resources also present for the biological macromolecules that we focus our attention on. Let's start by classifying them. There are types of them. Of course, in this presentation, we're going to focus on two types. One protein-based and the other one polysaccharide-based biomaterials. The most famous ones are collagen, fibrin, and fibroin under the heading of protein-based biomaterials. And if we try to understand which molecules are biologically important, and we can use them in the design of biomimicking disease organs or non-functioning tissues. Agarose, alginate, hyaluronan, chitosan, and pullulan are the most important ones. And of course, tissue biomimicking is very important. For that reason, extracellular matrix definition could be helpful for us to understand the characteristic, either biological, physical, and chemical properties of either the tissue or an organ that we're trying to biomimic. So extracellular matrix is very important because it helps cell to bind together, regulates a number of cellular functions. These are adhesion, migration, proliferation, and differentiation type of regulations and binding properties. So that's why extracellular matrix composition of them, understanding for what type of tissue, what are components of the extracellular matrix in the design of the either a scaffold or a fiber or a film is a very, very important point. That's why I try to emphasize it by changing the color into red. Why extracellular matrix is important. Okay, then what are the mimicking components, and how can I mimic extracellular matrix? Of course, we can categorize biomaterials as non-extracellular matrix components or an extracellular matrix components. Now, as you see here, cellulose, chitin, chitosan, alginate, dextran, fibrin are type of non-extracellular matrix components. But be careful, these are pure forms of them. If you modify or derive another form of them, then they might be extracellular component. But now here on this slide, we are talking only pure biomaterials, cellulose, chitin, chitosan, which are not derivative of any molecule. So extracellular matrix component that mimics, those are collagen, and there are different types of collagens depending on the tissue that you try to biomimic. Collagen types differ. Gelatin is very important. Fibrinolactin, laminins, elastin, glycosaminoglycans, and decellularized extracellular matrix. All these are the extracellular matrix components that can be used in the design. And as you see here, we can be able to group them under subheadings in order to repair damaged organs or creation of artificial organs. Organ culture systems are very important, and on this point, tissue engineering comes into our consideration, where tissue engineering is the interdisciplinary field that cells, culturing techniques, advanced biomaterial preparation, everything is included in. So organ-specific matrix, how to design an organ, either by using regenerative medicine adaptive technologies. These are there are a lot of different types of techniques to design it. Of course, drug delivery systems is also very important. Drug delivery is a rapidly developing field in biomedical research. It is interdisciplinary and requires expertise in biotechnology, pharmacology, microbiology, biochemistry, polymer chemistry, and material engineering. So all these interdisciplinary fields should work together, create a synergy, and try to find a solution for the real world problems. So growth factor, immobilize, substrate, all these can be able, scientists should be able to include all these for the scaffold and try to differentiate, for example, mesenchymal cell into different types of cells. So there should be a cell biologist in the team also, in order to be able to create artificial organ. Organ reconstruction techniques, of course, is also very important. Culture systems, as I said, nanofiber sheets, and scaffold processing technology. Now, in our Tissue Engineering and Biomaterials Research Center, we are working try to specialize on sick fibroin biomaterial. As everybody knows, sick fibroin is derived from the bomboximari cocoons, and it is an attractive protein polymer for most of the biomedical applications. Now, in this presentation, I try to summarize most of the researches that we carried on in our field. Now, this is the primary structure of the natural protein sick fibroin, and glycine, serine, and alanine are the amino acids which are most abundant to the on the primary structure of it. Sick fibroin protein are particularly promising for artificial organ design for drug delivery systems due to their unique combinations of biocompatibility, biodegradability, self-assembly, mechanical stability, controllable structure, and of course, morphology. As you see here, fibroin fibril is shown as a cross-section of it. Here, fibroin fibril, and 45% belongs to the glycine, 29% alanine, and 12% to the serine. And different cross-sectional areas are shown here from the fibroin fibril, and crystalline regions and also amorphous regions are shown. Now, purification of the silk fibroin protein from the cocoons, silk cocoons, are very simple. And as you see here, in the purification step, we can group them under three headings. One is the degumming, the other one is the dissolution, and then purification, which is a dialysis method. So after we cut cocoons into small pieces, By using 0.1 molar sodium carbonate, we can be able to decam and remove the serine amino acid from the system. And after all these gaming amino acid removed from the system, we use a strong electrolyte solution to dissolve these fibers that we dry at room temperature. By the way, we never dry them in the incubator around 60 degrees Celsius. We can easily dry them at room temperature. That's why I prefer to dry those fibers at room temperature. And then dialysis have been done with respect to the water, and purified sick fibroin obtained. These are the some micrographs of the raw sick cocoon, and decam one. As you see, they are morphologically different from each other. And structural function relationship of the sick fibers are listed here, where lightweight is around 1.3 gram per cubic meter, unique mechanical properties that they have high strength, elasticity, and tautness, and good environmental, biological, and chemical resistance they have. Now, biomaterials related properties of sick fibroin. We know that we can easily convert sick fibroin protein into different forms: nanoparticles, microparticles, films. We can be able to convert them different pore sizes, and by playing with the manufacturing production technique, we can be able to design different types of scaffolds. Either we graft them, and all these provide structural support for cells and tissue formation in the scaffold. Of course, all of the natural biomaterials are degradable, and biodegradability is also important in most of the biomedical engineering applications. And I want to show you this slide. I prepare it to show what type of works we have done in our research center. This is sick fibroin scaffold, which have been prepared by using freeze drying technique at minus 80 degrees Celsius. These are the films. I will explain them to you. They are semiconducting materials, which is very, very important in the integrated circuit designs. And these are sick fibroin nanoparticles. These are microparticles of sick fibroin and fibers. Of course, there are other types also that we will be discuss later. And these are the light microscope micrographs of the nanoparticles. Of course, they are not clear, but 0.2 micrometer, that means 200 nanometer, and 0.3, 0.5, so they're around 500 nanometer in size. Of course, later you will see that we will use different techniques to measure them. And Dr. Nanyat Kalam is one of our PhD graduate. Dr. Nanyat Kalam worked on in vitro MCF7 cell apoptosis analysis of carboplatin loaded sick fibroin particles together with me and Associate Professor Pınar Tülay, where we collaborate with the Faculty of Medicine in Near East University in this work. And we published Nanyak's thesis in Molecules, which is one of the high impact factor journal. Of course, we focus on breast cancer because it is the 5th leading cause of death worldwide, and try to find a solution for the chemotherapy that is commonly used directly for the management of breast cancer, but as everybody knows, it attendant adverse effects. And in order to be able to solve this, our aim in this study is to encapsulate carboplatin, which is one of the drugs used in chemotherapy, with fibroin by using ionic gelation method, and try to understand, analyze, characterize apoptotic effect of MCF7, which is one of the breast cancer cell, during in vitro studies. Of course, characterization of these fibroin encapsulated carboplatin microparticles have been done by using FTIR spectrophotometer, scanning electron microscope, master sizer, and biodegradation methods have been applied. And of course, at the end of all these characterization, we propose that a dose-dependent increase in absorbance indicating significant cell death with p-value less than 0.05, and the direct apoptotic effect of fibroin carboplatin microparticles on MCF7 was confirmed. This is one of the work that we have done, and Nanyak was able to receive his PhD with this work. Now the second one belongs to Dr. Chidi Enmekwo. He was graduated from the Biomedical Engineering, Near East University Biomedical Engineering PhD program, with the bioscaffold that he prepared to find a solution for the cartilage tissue engineering. Of course, the base is a fibroin. I want to give a short introduction related with the articular cartilage, which consists of specialized proteins and macromolecules. Of course, in order to be able to biomimic articular cartilage, we have to understand biological chemical composition properties, either physiological and anatomical properties of it. This is the way of designing, tailoring scaffolds or films or particles for the biological tissues or organs. Now, as you see, we know that proteins and macromolecules play a lot of important role, and joint friction reduce at the interface of two long bonds in a rigorous mechanical environment. And as knee, hip, and shoulder merely contain chondrocytes in a vascular structure, we're trying to understand where articular cartilage cells, chondrocyte cells, are available in the design. Design means the scaffold that we designed in this study. So cartilage tissue engineering is very important, especially the injectable hydrogels are important solution for cartilage tissue engineering. Uh, in today's medicine, especially personalized medicine, they use injectable hydrogels by isolating cartilage chondrocyte cells from the patient himself or herself, and within 48 hours they culture that isolated cells into the injectable hydrogel and inject that into the human patient's body. So it's very important, especially in the personalized medicine. Now, 3D scaffolds, production of 3D scaffolds. Of course, homogeneous distribution of deposited cells that are generated cartilage tissue are important, exhibiting excellent mechanical properties. So we have to characterize them by using mechanical properties of also, and enhanced extracellular matrix production is very important. Of course, for this study, GD tried to understand what type of biological macromolecules or natural biomaterials should be used to be able to mimic natural cartilage extracellular matrix. And we find out that during our research, curcumin, which is also naturally derived biomaterial, and it's a component of the golden spice turmeric, can modulate multiple cell signaling pathways. So, of course, in the past half century, all this has shown. And in keto and enol form of curcumin, if we check which one is better, enol form of curcumin is more important than the keto form in most of the therapeutic devices, either in the design of the drug delivery systems or controlled release scaffold devices. They use enol form. And the difference between the keto and enol, as you see here, we have a carbonyl group. Here we have a hydroxide group, and enol form is preferred. And some pro-inflammatory disease that use of curcumin yields promising results in patients are listed here. As you see, cancer, cardiovascular disease, arthritis, peptic cancer, gastric cancer, diabetes, acute coronary syndrome, beta thalassemia, chronic bacterial prosthesis, hepatic conditions, alcohol intoxication, a lot of different type of diseases can find solution by using curcumin. So what we try to do, we do not want to buy any isolated curcumin from the market. So that's why we collaborated with the Faculty of Pharmacy of the nearest university. And with this collaboration, we can be able to isolate and purify our own curcumin by using percolation extraction of curcuma longa, and then after the process of rotary evaporation, column chromatographic method is used for separating curcumin. Of course, I don't want to go into details of our work because of the restricted time. As you see here, these are the different types of hydrogels that Chidi work on, and we spent a lot of time to find out the optimum conditions and production parameters for these hydrogels. And after that, of course, characterization is very important. FDIR, SEM, scanning electron microscope, differential scanning colorimeter techniques, different types of characterization techniques have been applied. Of course, we sent our samples to either to Turkey, to BTEC, or Middle East Technical University. And at the end, as you see, different morphologies, different pore sizes, uh, different chemical and physical properties and also biological properties have been analyzed from the scaffold that Chidi produced. Now, of course, our aim is to inject these hydrogels into the biological system. So blood compatibility is very important for the implantable materials, biomaterials. That's why in vitro coagulation test analysis have been done, prothrombin time analysis, INR, APTT. So we try to understand with the contact of the blood what happened to the thrombogenesis mechanism, how the material, biomaterial or hydrogel with different compositions affect this mechanism. Of course, at the end, we find out that curcumin, unfortunately, negatively affect the thrombogenesis mechanism, but of course the amount of curcumin that you have to work on to find out the optimum value is very important. So this is one of our results that we have to take care of the amount of curcumin if we want to get compatible, blood compatible hydrogel at the end. And now this one is another work. All are naturally derived materials. We publish this work. Of course, this belongs to Leyla Karimizarandi. She's one of my intelligent and smart master students. Now she is in Iran now, and 2 weeks ago she sent me an email and asked recommendation letter to apply PhD. Of course, I feel very happy because Leyla should be an academician, and she worked very hard in this work. This is also related with the cartilage tissue engineering, but it's very interesting because in this work we use eggshell membrane, and it's very innovative because, you know, in bio engineering, waste materials, recycling waste materials are very important. And we published this work in International Journal of Biological Macromolecules that have an impact factor above 5. Eggshell membrane has natural extracellular matrix component within itself. So that's why these hydrogels are very successful on the chondrocyte cell proliferation. And as you see, the scanning electron microscopy, pores and morphology is completely different from the one that Chidi prepared. Here we have curcumin, sigfibroin, different formulations, I said, and of course it's a different type of trial. And but in Leila's work, we work with axial membrane, and as you see, it has completely different morphology. Now, articular cartilage, it is poorly cellularized, and it is the simplest tissue that it can be biomimic because it is non-vascularized connective tissue. That's why the first step in tissue engineering to find a strategy is to start with the cartilage tissue. Aim of our study is to check and analyze whether the hydrogel that we design can be able to proliferate activity of human chondrocyte cells. Again, I want to emphasize that we collaborate with the Faculty of Medicine of Nearest University with Associate Professor Rasime Kalkan, and that's why this work is very successful. Of course, characterization, X-ray diffraction analysis, FTIR, ICP, MSM, swelling kinetics, biodegradation, all have been done. And antibacterial properties also analyzed. And at the end, the result indicates that the attachment and cell growth of chondrocytes were examined by phase contrast microscopy and by MTT assay at 24, 72, and 7 days. These are the results. And we suggest that cartazine sigfibroin axial membrane hydrogel can potentially function as a promising cartilage substitute for tissue engineering applications. Of course, we have worked on different types of biofilms, graft form of sigfibroin, scaffolds, and as you see, these are the film forms. And of course, platelet adhesion test also applied for these films. The one that I show you is a sigfibroin graft polyethylene glycol methacrylate adhesion test. And oh, sorry, I'm so sorry. And we also published this as a sigfibroin as a non-thrombogenic material in International Journal of Biological Macromolecules. This is another work. Here, sigfibroin polypyrrole biofilm have been prepared with Dr. Eyüp Kavalcı in his master thesis. And of course, some micrographs were shown here, and electrical conductivity test applied. Our aim is to prepare flexible electronic semiconducting films for those type of applications. This is one of the master theses. Mitsubishi Moyo was graduated with this very successful master thesis. Here we work on tri-layer, bi-layer polyelectrolyte complexes. As I said, our aim is flexible electronic films, and these are 3-layer or bi-layer polyelectrolyte complexes. Now it is under revision, this paper. Chitosan, sodium alginate, and sigfibroin were used in the design of those polyelectrolyte complexes. We're trying to use natural biomaterials to improve the biocompatibility, that we can use these films in the implantable integrated circuit devices. AFM graphs were shown. These are surface analysis of those films, and a lot of characterizations have been applied. And of course, scaffold freeze drying techniques was shown here. Another work, different types of scaffolds, antimicrobial analysis of sigfibroin, ofloxacin scaffolds for different purposes. And another polysaccharide, which is very important one. One of my PhD student apply a pullulan polysaccharide to design nanoparticle drug delivery system for the Alzheimer's disease. It's a polysaccharide. I don't want to go into the details of its chemical structure. It is produced from starch by the fungus Aurea basidium pullulans, and it's very important. Dr. Gülcem Altınoğlu was graduated from our department PhD program. With this thesis, we published them, development of novel polymeric pullulan nanoparticles for Alzheimer's disease. She is neuroscientist, and she was graduated from the King's College London, and she did perfect work here. And our aim is targeting the cholinergic oxidative stress hypothesis targetable polymeric pullulan nanoparticles. And here, this is the chemical mechanism of the reaction mechanism, how we synthesize phosphorylated pullulan nanoparticles, and some micrographs of the pullulan nanoparticles. Of course, to give a targetability property, we use acetylcholine, because as you see, the targeting hypothesis in the Alzheimer's disease is the cholinergic oxidative stress hypothesis. That's why acetylcholine is used here, and as an active ingredient, rivastigmine is used, and these particles are only pullulan nanoparticles, but after the acetylcholine and rivastigmine encapsulation, these are the some micrographs of our pullulan nanoparticles. And of course, a lot of characterization have been done. I don't want to present them here because this is also under revision. Zeta Sizer potential analysis results are shown here to emphasize their nano size. As you see, pullulan nanoparticle, the zeta average of them is 33.34 nanometer. As we at acetylcholine, 34.06 nanometer. As rivastigmine loaded, 31.93. And acetylcholine with loaded rivastigmine is 32.65. So polydispersity index and zeta potentials are also indicating that these particles are also very stable. Of course, during the characterization, enzyme activity, a lot of characterization techniques have been applied. But I don't want to go into details after we publish. I hope I can and Gulcem can give you a seminar on that. Biomedical industry applications, you see, functional coating electroactive biomaterials are required. Autograft systems from natural tendon is another requirement. Tissue engineering approach is very important. Of course, silk fibroin, pullulan, gelatin, collagen, most of the natural biological molecules are important components of the biomimicking system. Intelligent textiles such as wearable real-time health monitoring systems and electrochromic systems, as you see, active, either active or passive ones. And now there are a lot of things, of course, I can present, but I would like to thank all of you for your patience with this embroidery from North Cyprus that have been done with silk fibroin cocoons. Thank you so much.
Thank you so much, Doctor, for very informative and interesting presentation. We really appreciate you putting this much time and energy to inform us about very interesting projects that you have implemented. This is very valuable, and now I thank you very much, and I give the room to Dr. Marziye Geranpaych. If Doctor, you're ready, we are. And if you may please, after the second presentation, we will have a kind of limited short discussion about the questions, the upcoming questions. So, and later after our second presentation, we will consider the questions. So, Dr. Geranpaych, the room is yours.
Okay. Hello, everyone. I hope you have. I enjoyed the previous presentation. It was so informative. Thank you. But my approach is a little bit more general. I'm going to talk about biomaterials. First of all, it's a good idea to look around and see all the efforts that all the engineers have done. When you take a stroll down the street and you spot one feat of engineering after another, but what you can't see all the time are the feats of engineering people are carrying around inside themselves. So, because engineering isn't just about what's around us, it's also about what's inside us. From medical implants that you can see here, just a minute. From implantations that you have inside the body or the cells that we are using, all the tissues that have been engineered and in different methods have been made, they are all the traces of biomaterials being used inside and engineers' efforts being used inside human body. From medical implants to artificial limbs and even bandages that cover your cuts, so many things have been designed from a special set of materials that work well with the human body. These biomaterials are vital to medicine, healthcare, and the world as we know it. Biomaterial. A biomaterial is a substance that has been engineered to interact with biological systems for medical purposes, that these purposes can be therapeutic or diagnostic. We have got a major science that is called biomaterials science or biomaterials engineering, which is the study of these biomaterials. And while we're studying biomaterials, we need to study and know about the elements of medicine, biology, chemistry, tissue engineering, and materials sciences. There are various materials that are candidates to become biomaterials, including metals, ceramics, polymers, composites, and even living cells and tissues. However, they are only candidates. So we cannot say that all the materials can suddenly become biomaterials. As long as they have the right skills of the job, they can be included. So to have the right, one of the factors that makes these materials a very good candidate for biomaterial is their compatibility with the biological systems, or as it is defined, biocompatibility to which is the behavior of biomaterials in different environments. And it's not only in their chemical conditions, but also their physical conditions as well. So, how biomaterials interact with the human body, and finally, how those interactions determine the clinical success of a medical device, defines the biocompatibility of that material. To check the biocompatibility of a material, we need to do various tests, including in vitro, or the ones that are done in the glasses, and also in vivos, which are done on different animals and organisms. And each of them have got their own advantages. So we first check the things, the materials, and whatever we have made in a glass to check whether the cells and the other issues have got the safety and they are fine, they're doing well. And the next step is the in vivo and the animal tests to further improve or investigate the characteristics of the material or the device that has been made. But there are four main ways a material can interact with your body. It can hurt you, it can dissolve and be replaced by cells. Your body can surround it with a protective layer, or it can bond with your living tissue. Biocompatible materials are the ones that result in healthy bodily responses. They don't tend to cause blood clots or infections and lead to normal, uncomplicated healing, which can be really important because for most applications, your biomaterials will at least partly come in contact with bodily tissues or cells, and often for long periods of time. This occurs. For example, it can be for months, years, or even for decades. Over years, the materials we consider biocompatible have changed a lot. From the animal tissue used by Egyptians to stitch wounds, to the wood that was used in older days as peg legs, and to the carefully engineered materials that are used today. They are all the history and the way that the materials have changed and the definition has changed. To get some examples, some of the more prominent ones today, I should mention titanium and stainless steel, that they are used from joint replacements, dental implants, and even braces. Another one that is used is polyurethane, that is used in catheters, artificial heart valves, and other flexible devices. And finally, hydrogels are there, which can be used in contact lenses. Each type has different properties that make it useful as a biomaterial. So here we are going to talk about different alloys that I have mentioned: titanium and stainless steel. For example, these two alloys are biologically inert. That's why they are given more specific attention to, which means they cause little or no reaction with nearby tissue. Although still your body may recognize them as foreign, and there still might be some issues. Titanium alloy is also, it has got some advantages compared to stainless steel. Since it lacks nickel, which nickel itself causes allergic reactions. So when we do not have nickel in titanium, it can reduce the allergic reactions. Also, it has a lower density. So an implant with which is a lighter, it means usually lighter is translated as much more comfortable. But both titanium and stainless steel have excellent mechanical properties like strength, resistance to bending, and durability. That's why they are used for things that tend to get a lot of wear and a lot of tear, like teeth and joints. And also titanium is used often to surround things that wouldn't be biocompatible otherwise. For example, a pacemaker, we can say. But there are still some issues. Sometimes you can change the surface of a material to do even better than just not causing harm. For example, sometimes implanted devices that come into direct contact with blood, like pacemaker that I previously mentioned, cause dangerous blood clots or narrowing of blood vessels as the person's body tries to heal around them. But scientists have found the coatings and modifications to prevent this problem. For this purpose, they have used collagen proteins and a blood thinner called heparin that could prevent those complications. There are still some early researches on them, but this way, these proteins that I've mentioned, collagen proteins, they can speed up healing, and heparin can prevent the clots. So that is the way they can be used, or they can be modified to have the best outcome. Polyurethane is another one that has got two parts of hard segments and soft segments. These soft segments causes them to become more flexible compared to the alloys previously mentioned, and therefore they may have a higher levels of elasticity. So they it makes them more a better suitable option to use in the heart valves. Also, they are durable and resistant to tearing, so they have got the advantages, and there are some more advantages compared to the previously mentioned alloys. Hydrogels are also polymers. They are hydrophilic structures, the same as other polymers, that they're having another advantage over them, which is the cross-linker. So there are some lines the other way, vertical to the previous ones, and which causes them to be able to hold larger amounts of water inside. How can we get the advantage? There is something called PHEMA or FEMA, that is a hydrogel that is pretty unreactive and supportive of biological processes. It is tough, it doesn't degrade easily, it isn't absorbed by the body, and can take on many shapes and forms. Simply, we call it Swiss Army knife of biomaterials. Think that this one can be used in coating a drug and delivering it to the right place. This is a drug that you swallow through your stomach. It enters your stomach, and the hydrogel is covering and it's around it, and the drug molecules are inside, as you can see. When it gets to the acidic part of the stomach, it absorbs the water, and little by little it becomes expanded, and finally it causes the release of the drug inside the body. Depending on the structure and modifications, we can define and make it to be released in the right place in the right time. Sometimes in the stomach it's the better option, sometimes in the intestine. Depending on the situation, we can modify all the drugs. So these hydrogels can be used in wound healing, bioadhesive, artificial skin and cartilage, sexual organ reconstruction, and vocal cord replacements. Another usage of these hydrogels is that when you have got a wound that is caused naturally, or it can be a cause of a surgery that you've already had, you can use these hydrogels in that place to accelerate and help the body to do the things that it normally does. It's not adding any options or any capability to the body. The only thing it does is that it's just helping the body to do whatever it is doing much faster, and therefore causing a healing to be improved much faster. Another thing, another usage a little bit away from the ones that I was saying is going to the nanosizes. This is the article I've been working on. It is modifying the drugs using particles or different proteins to modify a drug, bring it to the body. And as you can see, this is a process of metastasis. That a primary tumor is traveling and going all the way to a second side, and we are going to have the metastasis occurring. Through which it is proved that platelets that are normally engaged in preventing the bleeding, they get activated by the tumor cells, unfortunately, and they protect the tumor cells, helping them to migrate to the second place. What we did is using some particles to prevent the platelets to become activated and cover the tumor cells. And finally, when the tumor cell is not covered by platelets, the natural killer cells, or the good cells, white cells of the body, the soldiers, attack the cancerous cell and causes their death. Therefore, we can stop the cancer progression. Also, there are various particles that can be used. Using the particles that you have designed completely synthetic, or the ones that you can get them from the body, modify them, and then restore and enter them to the body. And one final thing I want to mention is that about the corona situation that we have got. There are a lot of vaccines that are being made, which I think we can consider them in this group. There are virus vaccines, viral vector vaccines, nucleic acid vaccines, and finally protein-based vaccines. There are the 4 groups of vaccines that are now being worked on to fight coronavirus, which is, we can think of them as Trojan horses. We bring them to the body, we activate the immune system, not really, I mean, it's just it's not a real one. It's something that we made and we entered the body so that the immune system is alerted. When it is seen and is in contact with the real virus, it can be protected. The final thing is that the safety of these materials that we use. We have to, as I said, first do the in vitro testing, then we do the animal or in vivo testing, and therefore, finally, we have to do some other more careful experiments to make sure that these devices or materials that we're using and we are giving them to the public, they are safe enough to be used. There are a lot of stories behind the ones that, although they have been checked, they have caused a lot of problems, even death for the users. So the safety measures should be taken really seriously. In this photo, you can see the biomaterials and dental applications of them, and it's my final slide. Thank you for your attention.
Thank you so much, Dr. Marziye. That's a very interesting topic, especially talking about the usage of biomaterials for the vaccines. I think everybody now these days are very interested in all, let's say, the issues and topics, all the discourses that is related to the coronavirus. And also, again, I would like to thank you, both our keynote speakers. We have kind of a very limited time, but I think if there is any question, or if there will be further discussions about the usage of biomaterials in different fields of the study, in different fields of expertise, I would like to ask all our participants to share their questions, to share their ideas and knowledge. So we are all waiting for your ideas, or if there is any question, you can ask the keynote speakers. Okay, I think there was one question. which Dr. Terin, I think you've answered that question.
Yes, I think so. Because Mustafa wrote that he thanks to us. Yes, thank you so much. Thank you.
Thank you so much. So if there is no further question or discussions, I would
Doctor, I have a question.
Sure. I would put my microphone on mute so you can discuss different issues.
Okay. Can I go ahead?
Yes, of course. Please go ahead.
Okay. I want to thank the presenters for the wonderful topics and the presentation. But there's an area I want to find out, though from our own field, these days we tend to pursue sustainability both in material usage. And I'm aware that stainless steel is applied in balustrades and balconies of houses. But what I want to find out is that if we're going to rate these materials mentioned, these biomaterials, let's say like the stainless steel and polyurethane, which of these has a higher rating in terms of sustainability? So if we are going to make selection in a building, because some of those factors may be taken into consideration. So I don't know if I'm asking out of this teaching, because it looks like the presentations are tilting more towards the medicals. So that's my question. Thank you very much.
Thank you. Yes, Professor.
Yes, Ercan, thank you for your question. But unfortunately, we are expert on the biomedical science. So application on the biomedical science, those biomaterials are more specific ones. But if we think about the architecture or construction materials, maybe antibacterial biomaterials can be used during construction. It might be helpful for the bacterial colonization, and it prevents those bacterial formation. That might be one of the application, but it's not the answer for your question. I'm so sorry.
It's okay.
Maybe those I know from old village life, they use some of the plant juice, mix them with the paint, and they use them for their house construction.
As pigment.
Yes, those might be interdisciplinary application with the biomaterials and your discipline.
Okay.
But of course, if we work on it, we can find a lot of applications. So it's a synergy. If we can be able to create a brainstorm, for example, when we first start to discuss biomedical science with the computer science, we couldn't understand each other. But later, you can find a way that you can apply that interdisciplinary field together, and it's a very creative one. So it's same for the other disciplines. So we are here. If you want to ask anything, contact details of us, and we can collaborate.
All right. Thank you.
You're welcome.
Also, there is another point for polyurethane. as Dr. Terin said, it's not our areas of expertise. But as long as I know, polyurethane is famous for its flexibility. Although it has got a UV resistant, it has got its own advantages. But compared to the stainless steel, which has a longer life and which is not even comparable with the other one, yes, polyurethane economically might be a better option, but stainless steel, its ability, its characteristics, and its strength is not comparable with the other one. In our major, we use it because of the flexibility that it adds to the characteristics, which is sometimes really vital. For example, as I said, for heart valves, that they need to become a little bit flexible. We accept that, okay, it's not as strong and as doesn't have the strength of the stainless steel, but still that is the advantage.
All right. Thank you very much.
No problem.
Okay, I have a question.
Sure. And I wanted to add something also. Biomaterial is my field of expertise. I'm an architect and a lecturer at architecture faculty. But from the architecture point of view, I think the very point that we can take advantage of biomaterial is the fabrication. So you know that sustainability is a broad issue, and in each step or in each stage of design, we can discuss this issue or this approach, we can enter this approach. And I think the very, let's say, collaboration, the very first collaboration that can happen is an architect finding the special characteristic that he or she is looking for a specific design. So to clearly identify the aim of design for what kind of or for what type of, let's say, fabrication. Is it the structural basis? It's a load-bearing system? It's a non-load-bearing system? Is it just going to be used as a flexible structure that allows the facade? For example, we have the kinetic facade structure that can breathe like the lungs of a human being. It can breathe, expand, and go back or contract back with the conditions of climate. So if we find the very aim and goal of architecture, and we ask, let's say, the biomaterial experts or bioengineers, with this collaboration, you can come up with a very interesting interdisciplinary research that is what these days in all universities, like all the fields, are collaborating with each other, and they're looking for these types of collaboration. So it's just as exactly Dr. Terin and Dr. Marziye mentioned, you have to find the boundary between these two different, apparently, seemingly, these two different fields of the studies.
All right. Thank you very much.
Thank you.
Sorry, I have a question. First, I want to thank all of lecturer for this great presentation. I have a question and point to share. The question is, what is the characteristic, like for example, biocompatibility? And I want to ask for another characteristic we can focus when we study these materials in the biological system. The point I want to share is about the calibration also. From engineering view, I think the best view for engineering is to study the materials from physics, from mathematics, from the math. This information is good for engineering part, and understanding more the engineering part of these materials, like the physics equation, the math. This information is good for us to make simulation for make engineering part. That's it. I want for the question answered.
Yes, Maram, thank you for your questions, but would you please repeat your questions one by one that we can be able to answer?
The question is the characteristic we can study it. For example, the biocompatibility. So what else we can focus in the human body when you study the materials in human body?
Yes, you know that mechanical properties are very important, stress-strain relationship. So it depends to the either the hard tissue or soft tissue that you are dealing with. So I think that's the point that you ask. Am I right?
Yes.
Yes, of course, on engineering point of view, mathematics, physics, all these are very important in the modeling, as we discuss in our lectures.
Yes.
So in biomechanics, you know that in we have to be able to use Monte Carlo simulation to be able to understand the biomechanics of it. So yes, those are also very important. But depending on the aim of your study, for example, if you're one of your aim, first of all, you know, in biocompatibility, we have at least 8 parameters to be able to characterize for the biocompatibility. Cytotoxicity, acute systemic toxicity, blah, blah, blah. So we discussed that in our course, but it's not possible for all of them to be applied. So that's why if you modify the biomaterial, natural biomaterial, either by grafting or cross-linking, or branching, you have to characterize it chemically, first of all, whether that grafting occur or not. And when you approach to the end product, after you approach, you have to check mechanical properties of it. Maybe if it's a soft tissue like skin, you have to check elasticity. All right? So it depends to your research problem. Thank you.
Okay, thank you.
Okay. Thank you.
I think Azmi Hoca also has question. And Valerie also. Azmi Hocam? Yes. Who? No. Mustafa raised his hand. Am I right, Mustafa?
Yeah.
All right. I confused.
Yes, my name is Mustafa, one of the students in pharmaceutical botany.
You can ask your
Thank you,
question.
Mustafa. Thank you.
All right,
Please share.
my questions is about if there are any properties that we take into consideration to avoid like tissue rejection in human biosystem. Yes. For example, incorporating another entirely different tissue or material from other living things, for example, a plant. Is there any tissue rejection? And if there is any, how can we overcome that?
Of course, there are tissue rejections. Our aim is by applying surface modification to the biomaterial, depending on the research problem, we have to minimize the tissue rejection. This is our aim. So that's why blood compatibility anticoagulation experiment is the first biocompatibility experiment that we applied for the biomaterial. It's a very important point.
And the second question is, having known all these characteristics or haven't tested it in a living biosystem, the next thing is the differences in asynchronous growth. For example, if we incorporate certain materials into the tissue of a living things, for example, human, like to be specifically a bone, for example, the osteocytes or the cells of the bone do grow, for example, in children. How can we synchronize the growth of this living tissue as well as the increase in the, as well as the feature of the incorporated biomaterial?
Mustafa, am I going to answer the question? All right, let me answer you. Now, in vitro, would you please mute your microphone? Now, in vitro analysis, and ex vivo analysis. So these are the ways of analyzing. If you mute your microphone, it will be better, Mustafa. Yes. Now, in vivo analysis is the one that you have to you have to give a chance for the biomaterial to contact with the living system. So, so that is the one way to understand sensitization, irritation, properties of the biomaterial, and the rejection also. But now, new trend in the tissue engineering, in the regenerative medicine, is to use microfluidic devices. Instead of using animals, they design by mimicking, for example, artificial lung within a microfluidic device, and they test the biocompatibility within that. So these are new trends in the research. But of course, they are all very important, and depending on your research problem, you have to design those type of experiments. Rejection is one of the important problems that biomaterialists should solve. Thank you.
Thank you, Dr. Terin. Thank you, Mustafa, for your question. Would you like to continue your question, or? Okay. Dr. Marziye, if you would please add your opinion on this matter.
I think Dr. Terin's explanation is very complete and sufficient. It's always the same. We have got in vitro and vivo, as I said, different experiments to check it in different steps, whether a rejection, a biocompatibility, even biodegradability of the material should be checked. So every step has got its own processes. Still, when we use it in human body, there are sometimes some special problems and different issues that can occur. They should be resolved. It can be either by changing the polymer we use, the I don't know, different amount of the drug we use. The coating can be changed. There are a lot of criteria, characteristics, and things that can be I mean, always optimized when we are working with a living thing, I can say. So it all depends on the material we're talking about and the step that we see the problem, and a lot of things are involved in the situation. So there are various ways to solve the problem. Thank you.
Thank you so much, Dr. Marziye. I think, Ejing, you have. Would you like to share your opinion because you raised your hand?
No, it's not me. It's the person whose hand is up here is Gules
Valerie.
Valerie has a question.
Yes, Valerie, would you please share your opinion or question? Thank you.
Okay, thank you. I want to first of all thank the presenters for their very nice and illustrative approach of the information they gave us this evening. So much thank them, and I really benefited a lot. But at the level of my question, first of all, has been answered by Dr. Terin, even without knowing, because I had a question in mind to ask. Now we have talked about the different the classes of vaccines. After talking about all of them, now at the end, Dr. Marziye mentioned that we have to verify, like we need to really do the testing to be sure that they are compatible, and mentioned that we can use animals and so on. But Dr. Terin came in and answered that you can prepare a system, a microsystem, because I wanted to propose that instead of using animals again, we can use our background knowledge in medical micro and nano systems to produce something like lab on a chip or organ on a chip to tackle this kind of issue, not to use an animal. But Dr. Terin has already answered it, so thank you so much. I think I don't have any other question.
Thank you, Valerie.
Thank you, Valerie.
I still have to add that a lab on a chip is not complete, and although it can minimize the number of animals we may sacrifice or use during our experiments, still it's not a complete model of a living organism. There are a lot of, I don't know, organs involved that, okay, in a lab on a chip, you can just make the model of different organs and tissues and see how they affect each other or how the drug affects each tissue. But still, a real organism must be used, I believe, before using a drug on a human to make sure that all the organs together are working and are being safe to continue the drug and usage of the drug in a real situation.
Thank you, Dr. Marziye. Our next question is from Gulce. Sorry if I'm pronouncing your name correctly. Okay. I think all the questions have been answered. If anyone would like to share the opinion or any question, we still, I think, have 5 minutes. Actually, no, we have passed the seminar time. So I would really like to appreciate Dr. Terin and Dr. Marziye for putting too much time and energy for this seminar series, which was very informative and interesting for everyone from all the faculties. I really hope that this would be an introduction and a basis for further collaboration between different faculties. I hope you will have a very nice evening. Thank you for participating in our seminar.
Thank you, everyone. Good night.
Thank you.
Thank you, Doctor. Thank you, everyone. Have a very nice evening and night.